This is a production of Cornell University.
Okay. Thank you, Jason, for that introduction, and thank you, everyone, for coming to listen to my seminar. This is a partial completion of my B exam as well, so it was incredibly special to me. As mentioned, my topic or my seminar is Broadening the Base of Apple Genetics, a study of traits using Malus prunifolia. So before we really get into the meat of my project itself, I wanted to give us some framing information about apple breeding and how we got here in the state of the world of apple genetics and breeding. The apple center of origin is thought to be in Kazakhstan in the Kazakh mountains, with a population of medium to large fruits in these mountains, with early hybridizations from Malus baccata and Malus asiatica, which are thought to be possibly originally isolated groups of Malus seversii, which is one of the founding species. Humans then carried these larger fruited Malus seversii along the Silk Road, resulting in further hybridizations with possibly some other Asian species, such as Malus prunifolia, until arriving in Europe, where there was a late, intense interspecific hybridization with Malus sylvestris, which is a species known to be firmer and more acidic, but smaller in fruit size. Later, more intentional selection and growing resulted in the Malus xdomestica that we are more familiar with today, which is known to be firm, sweet, aromatic, tart with larger fruits. So that story of domestication, and there is sylvestris. So that story of domestication then led into hundreds of years of intentional apple selection and breeding by humans to bring us here where we have amazing cultivars such as Snapdragon and Ruby Frost, New York 1 and New York 2, which were released from Cornell rather recently. Apple, if you're unaware, is a clonally propagated species, and fruit is marketed by cultivar. This has resulted in certain cultivars becoming more widely planted and more widely sold. And these cultivars mostly have larger fruit with lower acid, higher sweetness, and are often red in color or have a blush. How we market and sell apples by cultivars have for commercial use has resulted in a reduction in diversity of apples that are grown for commercial use, though breeding pools and repositories like the one in Geneva show higher diversity still, so we have not lost the diversity of Malus in this breeding efforts. Here we have in the graph, we have the top 9 cultivars that are sold in the US. You are likely pretty familiar with these cultivars, Granny Smith, Pink Lady, Golden Delicious, Gala, Red Delicious, Honeycrisp, and Macintosh, to name them, and Fuji. These first degree relationships are demonstrated by these gray circles, and that just means that they are otherwise a sibling, a parent, or a child of these very popular cultivars, and there's plenty of hybridization between these as well to result in a rather reduced amount of cultivars commercially available, and this has resulted in some problems, specifically related around disease and disease resistance. Which brings us to my project. So Malus prunifolia is a West Asian species of apple that was possibly involved in domestication along the Silk Road. Depicted here in a photo, it would be on the smaller Malus species on the left of your screen. And then you can see here on the right a diversity panel of all of the Malus prunifolia inside of the genetic repository in Geneva. And you can see that Malus prunifolia as a species really runs the gamut in sorts of fruit shape, color, and size. And so it's a rather interesting species. The specific Malus prunifolia that I have used in my study is Malus prunifolia 19651, which from here on out, I am just going to refer to as Malus prunifolia because I will trip over those numbers constantly if I don't. This individual has been used as a part of the Malus core collection, which means it has been included in multiple linkage studies and mapping studies. And as an individual, it clusters with other Asian species, but not always with other prunifolia accessions. And its dihydrochalcone profile, which dihydrochalcones are phenolic compounds that are of interest in increasing beneficial health compounds in apple. Its dihydrochalcone profile indicates that there was some species admixture in its background. So we're not even sure that it's actually Malus prunifolia, but it is the designation that we are currently using. And it's of interest because of its apparent strong fire blight resistance and there's a thing, and other fruit biochemical traits, as well as its usefulness as a wild species in genetic studies, trying to understand novel traits from not underutilized species in breeding. The other parent in this population of trees is called New York Co1. Co stands for columnar, and it is a Malus xdomestica, meaning it is the standard domesticated apple that we are familiar with. It has columnar habit, meaning that it grows very vertically upright with short spurs and very few outward branches. So it's more like a pillar. I have some photos of it coming up later. And in its background, it has Panova, which is a German cultivar, Suncrisp, and Widgeck Macintosh, which Widgeck Macintosh is the origin of the columnar habit in Malus. And it is a large fruited individual with yellow skin and a slight blush to it, and other characteristics I will be listing later. So these two individuals are quite different, which results in a rather interesting display of trait segregations that I will be touching on. So as a general summary of this population, like I said, it was a cross made between New York Co1 and Malus prunifolia. And it resulted in 600 seeds that were then germinated. A number of them died due to a lethality gene, and resulted in a validated population of 423 individuals, which I validated using GBS markers and PCA, principal component analysis. And that broke down into 226 standard trees and 197 columnar trees, which is about 50-50, which is what we would expect in the segregation ratio. And then there's dwarf types within that. I'm not really going to be touching on trait architecture during this talk, though there are some interesting components of that I will be discussing in publications and findings later. So briefly, this talk is going to focus on three things: fruit storage, and I will be talking about storage genes and genotypes, associations, and then an RNAseq experiment that was used to clarify kind of what's going on in storage. Fruit organic acidity, genetics, pH and titratable acid was measured, associations were found, and then I'll be discussing deviations and distortions unique to this population or unique to this breeding pool in general. And then lastly, fruit quality traits, which I will just be touching briefly on fruit size. However, I measured a number of other traits, including Brix, presence of russet, and phenolic compounds, along with several others. And isn't this such a pretty image? They're siblings and have very different colors, flowers, but they're both columnar. This population is really stunning in flowering and fruiting, just in kind of the breadth that it covers, and it's always fun to walk through. So I followed this population for 4 years, 3 pretty intensely, and then 1 with some extra data to get 3 years to report on. So in 2019, 176 individuals fruited. The traits that I collected are listed there, including total acid, pH, Brix, phenolic compound, shape and size, color, and then fruit and cuticle disorders. 2020, 288 individuals fruited. And all those previous traits were collected, as well as mass. And then I added a color rimmer to the technical mix so I could measure more accurate color. 2021, 306 individuals fruited. And RNA samples were taken along with all of that previous data. And then I checked the population for starch iodine index at harvest. 2022, 308 individuals fruited. And I continued collecting data for storage time, fruit size, and then external characteristics that I was seeing. So in order to do any sort of genetic study, we need to have genotyped the population. So this population was genotyped using genome by sequencing, or GBS. and a collaboration between Cornell, the USDA, and Dalhousie University. It was then aligned to the Golden Delicious double haploid reference genome, and I used a principal component analysis depicted here on the left to identify and remove any outcrosses. So the 2 points on the far side of the charts are the 2 parents, and the cloud in the center are all the progeny. The 22 outcrosses were identified and discarded, and then a further outcross was found in genotyping later on and was also discarded before analysis. The filter settings I have are there, and it resulted in 51,442 sites, which is plenty for a genetic study. And so my genetic analysis has been pretty robust, which is exciting. So the first trait that I'm going to talk about here is apple storage. And in the first year of fruiting, this population only had like 22 individuals fruit in 2018 when I first got here. But even in that small subset of individuals, there was an apparent segregation of long storing fruit that stayed well in the cooler, on par with Malus domestica, and short storing fruit, some of which senesced on the tree and some senesced very quickly in the cooler. And I have quite an interest in reducing food waste, and so I found this trait to be of great interest. It's also important to note that since this is an interspecific population, and because of what we know from domestication studies in apple, fruit firmness and the ability to store fruit appears to be a trait that we selected upon in the early days of domestication and fruit breeding. And so adding in this wild species gives a kind of novel chance to look at some of these domestication genes and what we should really consider as we're working towards using wild species in future apple breeding. So my hypothesis is listed there. Briefly, genetic differences are causing differences in storage time phenotypes, and then my questions are listed there. What genes are causing these differences, are external biotic effects causing them, and really what effects are these genes having on the fruit over storage time. Storage is intensely researched in apple, and since apple is a clonally propagated crop, most of the research has been focused on modifying the environment surrounding the fruit, whether in growing or in storage, and not as much focus has been given on genetic causes. And that's mostly because once a cultivar is released and becomes popular, we can't retract it to fix an issue in storage. And so we work on modifying the environment around it. However, I really wanted to look into the genetic causes of storageability that I'm seeing in this population. So here, if you've been in any sort of intro storage class, you see a ripening curve for climacteric fruit, which apple is one. You can see the trends in respiration and growth, as well as the interplay with ethylene, which is a plant growth hormone that is heavily tied to plant respiration, plant growth, and then fruit ripening. With this understanding, I then looked into genes that had been previously identified in other studies in relation to ethylene as well as storage. The ethylene biosynthesis pathway is depicted here with the genes of interest that I'm going to talk about in blue. ACO and ACS have been identified in apple and somewhat well characterized. These genes have been identified and known to affect ethylene production over storage time. And so here's more information. ACO is located on chromosome 10. It is the final step of ethylene biosynthesis and has been associated with increased ethylene production. For a long time, ACO was considered like not that much of a factor in storage, but more recent studies have indicated that it is more of a rate limiting step than previously thought. ACS is the first step in this two-step pathway and has previously been reported as the rate limiting step. It's located on chromosome 15 and also is increased with associated with increased ethylene production. But first, before I got into that, I wanted to look to see if there were any biotic causes of these storage differences in this population. So blue mold, which is a Penicillium, is the it's the number one post-harvest pathogen in apple storage. And so I created a subset of the population half of them being long storing individuals that I had tracked at least a year previously, and half of them being short storing individuals, and tested them for blue mold resistance by stabbing them with a nail and putting Penicillium inoculum into that wound, and then placing them into an inoculation chamber, also known as a large plastic tote with wet paper towels to maintain humidity and ensure inoculation and sporulation. And from that, no complete resistance was observed. So all of the fruit did get infected with blue mold. And however, there was some slight variation in time to sporulation, but not enough to be significant. As mentioned before, ethylene is very important in fruit ripening and senescence. So the same subset was brought down here to the Watkins lab with Joseph and Chris Watkins to be measured for internal ethylene. So a measurement of gas was taken from the internal seed chamber. And from that, no real pattern of was found between the two classes. So no pattern in long storing and short storing. However, this is likely due to the fact that the fruits, there was ripeness differences in the fruits caused by its interspecific background. I can't use standardized ripening metrics like starch iodine index and penetrometers because the fruit are too small for a penetrometer. Some of them will get crushed just by the distance of penetration, and variation in starch has been observed in wild species. So I couldn't reliably use starch iodine index to quantify ripeness either. And also sometimes the seed chamber was simply too small to accurately draw gas out. So ethylene is still a mystery, but blue mold is not the cause of this resistance or this storability. So from the 3 years of data, I looked at segregation ratios, and they are depicted here. They are approximately 50% or 1-to-1 segregation ratio, which suggests a single large effect gene, which I then ran a genetic association using Tassel and then R to plot the results, which are displayed here as a Manhattan plot. And you can see there's a very clear and strong peak located on chromosome 10. And this peak co-locates with the region that has been associated with ACO. So to see if anything else was going on, I then genotyped the two parents for ACS and ACO, the two genes involved in the ethylene biosynthesis pathway, and found that in ACS, both parents, New York columnar 1 and prunifolia, were homozygous for the long storing or the low ethylene producing alleles. So ACS does not seem to be of any effect here, which makes sense since we did not see any peak on chromosome 15. However, ACO, the Malus domestic parent, New York columnar 1, has the two low ethylene producing genes, but in prunifolia, it has one copy of the low ethylene producing and one copy of the high ethylene producing. This means that the progeny will be segregating a 1-to-1 ratio between homozygous low ethylene producing and heterozygous. So one copy of long and one copy of low ethylene and one copy of high ethylene producing, which also validates this Manhattan plot, this genetic association. But I wanted to understand what was kind of going on in this population or in storage over time. And so I outlined an RNAseq experiment that used 4 long storing individuals, which included the long storing parent, and 4 short storing individuals, which included the short storing parent, which was prunifolia, and had collection dates that were connected over physiological time of senescence. So in short storing, there was 3 time points that were measured: a initial time at harvest, a midpoint, and then right before senescence. And then in long storing, there was 5 time points that were collected, which had all of those plus 2 additional harvest time points just to further give clarification on that gene expression curve. And all of these individuals in the study were also validated with their genotypes. So the short storing individuals had the heterozygous genotypes at ACO, and the long storing individuals had the homozygous low storing or low ethylene producing genes at ACO. This is just kind of the workflow of the RNA-seq that I did. It resulted in an average number of reads being over 3 million, the percent of uniquely mapped reads being at 81.43%. And I had 30,000 out of 52,000 of the total annotations that were mapped to the peel. So I decided to take tissue samples of the peel because it has been shown to be more biosynthetically active than the rest of the fruit during storage. And also the fruit tissue itself contains lots of sugars, which cause problems for RNA extraction. These numbers are a little bit low for a lot of RNA-seq experiments, but for a 3' profiling experiment, that's what this is sufficient to give us an idea and indication of what is happening over storage and time. So from those reads, a PCA was made to demonstrate the separation between the classes and see if an unsupervised WGCNA was able to be done. So here you can see across PCA1, which is the bottom, there's pretty good separation over time. So early collections mapped on the right side of that chart and later collections mapped on the left. And then across PCA2, we can see a pretty clear separation between the classes. So again, long storing individuals are blue and short storing individuals are red. So this gave us indication that we could use unsupervised WGCNA, which then created this cluster chart. The color names on the left sides of these boxes don't really mean anything other than names of the clusters, so you can ignore that. But these are groups of eigengenes that appear to be associated with each other. So red means that the genes in the clusters are positively correlated with each other. So as eigengene expression of them go up, other eigengenes also go up. Blue is negatively associated, so same pattern in reverse. And the contrast of this was between the two classes, so long and short, and the different columns here are group. So just comparing between long and short individuals, time, which was days, since each accession had different storage lengths because they were mapped to their individual senescence, and then time group, which was the 0, 0.25, 0.5, 0.75, and full senescence discussed earlier. So from that, these clusters were then made to create to demonstrate eigengene expression patterns over time. So the y-axis is the modeled eigengene expression. Higher means higher expression, lower on the chart means lower expression. And then the x is just that time point series. So as you can see, the short individuals in red only have 3 collection points and the long individuals have 5. And I have these two highlighted just to demonstrate kind of what this sort of output tells us. So on the far right, we have turquoise and blue, which you see that the two classes appear to be relatively similar in shape across time. We can assume that that is standard biological process in storage. And so I'm not going to be looking in that cluster for anything because they're both going through similar expression levels. And then on the left side, we have yellow and green, which have quite different levels, similar shapes, but different quantities of eigengene expression. And so yellow and green might be an area that I would look into for some causal genes or some pathways of what could be going on. And here's all of the clusters that I could potentially look into. The parentheses indicate how many genes are located inside that cluster. So some are quite large at 41,000, and some are quite small at 65. However, we don't want to just rely on these algorithmically decided expression patterns in order to explain what we're seeing. So with that information and with the association that I have discussed previously, I looked into the annotation files that were available for ethylene-related genes, and I found over 300 genes, and that included transcription factors, pathways, signaling path repressors, and synthesis pathway genes. From that 300, there were 30 that were differentially expressed in this pattern, and they were distributed across 7 of those previously pointed out modules. And from those 30, I found some of the most highly differentially expressed, so log fold over 1.8 and a p-value over 0.005. And from there, I went back to this biosynthesis pathway. And as you can see again, ACS and ACO there are the ones that we have understanding about. And so I looked at ACS. This again has no segregation in the population, and the gene expression or the transcript expression of that gene is not significantly different than between long storing and short storing, which you would expect because there's no genetic difference, right? In ACO, which we know there's a genetic difference, we would expect there to be a different expression, but again, there's no significant difference between the two classes. However, I do want to point out here that the causal mutation of ACE that causes the low ethylene producing and high ethylene producing phenotypes at ACO do not change the transcript level. It is a functional mutation, a missense mutation that causes the protein resulting from the transcript to function less. So we wouldn't actually expect any transcriptal differences at ACO. So what are we going to look at now? Because that doesn't make sense anymore, right? So I looked at other associated genes and other associated proteins and found this gene, SAM-DC. SAM is a pretty ubiquitous protein in biosynthetic pathways and in ripening. The DC is decarboxylatic. I always have to look. Decarboxylase. But SAM-DC has been shown in ripening studies to be upregulated by the perception of ethylene. So SAM is increased. The expression of SAM-DC is increased when the fruit or the tissue is receiving ethylene and starting the biosynthesis pathway again. And here we do see a significant difference between the two classes, where long storing individuals have quite a bit less transcription of this SAM-DC gene, and short storing individuals have an increasing expression of it over time, which makes sense because in theory, ACO is producing more ethylene in short storing individuals than long storing individuals, and then the fruit are responding to this higher ethylene concentration around them. So to look more into this, I then looked at the ethylene receptor and sensor genes, which do similar things. They sense ethylene and then start the ethylene senescence pathway. And in both of these cases here, we see a difference between short storing and long storing individuals, with long storing being significantly lower than short storing individuals. And continuing on that, these are ethylene response factors, which they are genes that get turned on when the senescence pathway that is controlled by ethylene kind of overproduces. And so they try to repress that pathway. So we would again expect these to be higher expressed in tissues that are sensing increased levels of ethylene. So I have highlighted here this response factor on chromosome 15, that you can see a very clear difference between the two types. And then here on chromosome 12, on the side of the screen, there are also two other ethylene response factors that show a difference between the two classes of fruits. And I really wanted to highlight this one at the top because the long storing individuals have almost no transcription of this ethylene response factor, while the short ones have transcription of it. So to kind of wrap everything up together, the biotic factors that I looked at, blue mold and internal ethylene, really didn't give a lot of clarity, but I was able to map an association that co-locates with the ACO region over 3 years. And then when looking at the RNAseq data, we see that the fruit itself is responding to an increased level of ethylene that is caused by the ACO gene. And that is backed up by the SAM-DC gene, as well as the receptors and response genes, which are higher transcribed in the short storing individuals and lower transcribed in the long storing individuals. Oh, before I move on. And then again, just to reiterate, this is of interest and importance in interspecific breeding because we need to do some interspecific breeding to introduce new traits, but we don't really know what negative traits are being brought in. And so this could be something that we have to pay attention to as we do that work. I am going to try to quickly go through stuff because I only have 5 minutes. So in apple, acidity is one thing that has been researched quite intensely. MA1 and MA3 are 2 genes that are reported to control the majority of malic acid accumulation in Malus. MA1 is reported on chromosome 16 and MA3 is on chromosome 8. On the right, there's a very short list of other genes reported to influence malic acid, though there is plenty more beyond that. I will just be talking about MA1 and MA3 here. So in this population, there's quite a distribution between acidity between the 2 parents. So this is data from 2021, though the patterns for this hold true over all years observed. And you can see that New York columnar 1 has significantly lower acidity than Malus prunifolia. CO1 comes in at 3.27 grams per liter of titratable acidity. And prunifolia comes in at 24.44 grams per liter, which, just for reference, apples are kind of thought to be somewhere between like 5 and 10 grams of titratable acidity per liter. So 150, New York columnar 1 is actually significantly lower acidity than kind of standard apple. And then prunifolia is quite a bit high, screamingly high acid. And all the progeny fall within these 2 parents every single year. So great candidate for genetic association. Because the knowledge of these 2 genes, the population was genotyped for MA1, and columnar 1 was found to be homozygous dominant, which would indicate that it has a high acid phenotype, which doesn't quite make sense. Prunifolia was found to be heterozygous, meaning it has 1 high acid allele and 1 low acid allele. So from that, we would expect a segregation ratio of 1:1 of homozygous dominant and heterozygous. However, we find an actual segregation ratio of 1 to 1.58, and that is due to that lethality gene that I mentioned at the very beginning that is linked to MA1. MA3 was also genotyped, and CO1 was found to be homozygous recessive, meaning it has a low acid genotype, and prunifolia was found to be heterozygous with 1 copy of high acid and 1 copy of low acid. And again, we would expect a segregation ratio of 1:1, and that is what we observe in the population. From that, I again did a genetic association using Tassel and GLM, and found this peak located on chromosome 8, which co-locates with the MA3 region. It is interesting to report, though, that we really don't see the peak on chromosome 16 that we would expect with MA1. We do see it slightly in the pH chart, but that peak is inconsistent over years, and it's never found in the titratable acidity, even over all 3 years. And it shows up in pH 2 out of 3 years. And I just want to point out, again, the ranges of data. Titratable acidity goes from 3 to 24, and pH goes from like 2.6 to 4.8. And so it's a lot easier to create kind of false associations without that granularity of data. And so from that, it brings up the question of what is going on here. The primary acid in Malus is malic acid, which we assume is about 95 or upwards of 95%. However, it's been found in wild species that secondary acids such as citric acid accounts for up to 60% of the acid profile. So to check if citric acid is what's causing this wide discrepancy in acidity, I tried to measure citric acid or the acidity profile and do associations with specific acids. Initially, I tried an enzymatic assay, which was inconclusive. I then moved to HPLC, and in HPLC, I found no signal, or it was an unreliable signal that I could not replicate from day to day. And so I sent a small subset to Laling to check for, or to use GCMS to check for citric acid. And there was no citric acid signal observed, but there was a wide variation in malic acid in the subset, which validates the high distribution that I was showing earlier, and also accounts or informs us that the majority of this acid is malic acid, and we can assume other acids are not really playing a part. So, like I said, the acidity profile is not due to these secondary acids, but we know that MA3 controls the majority of acid expression in this population, which kind of indicates that MA1 seems to be non-functioning in this population, or it's under repression. New York CO1 has high acid alleles, homozygous dominant for the high acid, but the lowest acid phenotype in this entire population. So this is a bit outside of this dissertation work, but in order to identify kind of what's going on or give indication, in conversations with Dr. Susan Brown, she mentioned that MA3 seems to control the majority of the acid in her breeding populations. And so MA1 could be sequenced to identify potential allelic variations that have lower expression or lower controls. And then we could identify the source of that allelic variation to see if it's unique to the populations here or unique to some of the background of New York columnar 1. Lastly, and I'm going to try to speed through this so I don't go way over time, fruit quality. Again, I measured all of these traits, but I'm really only going to report on fruit size today because it has a clearer story. So I measured fruit size as fruit height, fruit diameter, and fruit mass. And here are the distributions for 2021, which again had just the most robust data set. So that's what I'm reporting on right now. And you can see again that the parents are on each side of the chart, with New York columnar 1 being the large fruited one and prunifolia being one of the smallest or the smallest in the population, with a distribution of progeny between the two. So using that data, I created another genetic association with these 3 traits, and interestingly found peaks on chromosome 13 in both diameter and height that the peaks were located at the same SNP, so 176952. And the peak that is on chromosome 13 in mass is very nearby the SNP as well. This is of note because no fruit size loci have been identified on chromosome 13 for apple yet. There was also peaks identified in this association on chromosome 5 for fruit height, chromosome 2 for fruit diameter, and chromosome 3 for fruit mass. So briefly in summary, I talked a lot about fruit storage and what is going on behind the scenes over storage time. I presented a wide variety in fruit acidity and genetic associations and controls in this population, and then also presented some data on fruit size. And all of this together kind of tells a story of using interspecific breeding in apple breeding, and also traits that could be paid attention to as we're doing that, and also somewhat goes against the standard idea of interspecific apple breeding, which is that all interspecific hybrids are small fruited and high in tannins, high in acidity, low in sugar, all these negative traits. And this project kind of tells a story that in interspecific apple breeding with careful parental selection and some patience, you are able to really get some decent sized and decent tasting apples after one generation that could be then used in future breeding with some of these useful traits. I'd be remiss to not acknowledge all the people that have helped in this project and in my research, namely Dr. Susan Brown and Kevin Maloney in the apple breeding lab in Geneva, who their mentorship has been invaluable to me. All of my collaborators, my committee members, the current lab that I'm in Geneva with Jason Londo, and then the grape breeding lab, which have been great friends and collaborators, and my funding through Bowler Grant and Schmidt Novak. And then none of this happens in a vacuum. And so I want to thank and acknowledge all of my friends and family that have supported me through all this and kept me sane as I'm staring at apples for 16 hours a day. And with that, I think I'm done and I would take any questions. Marvin?
Quick question. So storage is a human construct, it's an artificial construct, and yet in these wild populations, you're finding variation in genes that impact storage, which wouldn't have existed when they were evolving and so forth. So what in nature is selecting for some of these differences in genetic traits that are shown up in storage? Like what's the selection pressure in nature when you're looking at these wild species? Is there like an optimal time for these apples to hang on a tree before they're consumed, or
Yep.
you know what's going on there?
Yeah, I mean, okay, so the question is, like storage is a human construct. And so during domestication, how were these storage related genes selected upon, right? Okay. And I think the answer is they weren't necessarily selected on for storage. They were selected for the ethylene senescence effects. So in this population, some of these trees senesce on the tree. And so if you think in nature, those senescing, rotting fruits are not very useful to animals because they're breaking down, the sugars are breaking down, the starches are gone. And so as animals were consuming them, they likely selected for ones that did not rot immediately, and then the seeds spread. And then as humans moved them along the Silk Road, probably something more akin to storage was selected for, just movability and transportation of fruits. You don't want your bag to be full of rotten apples, and so you would discard those and you would keep the ones that stayed sound over time, right? Yeah. Yes.
Yeah, thank you for the presentation. How did you select the storage period for how long, between like short and long-term storage?
So the question is, how were the classes defined between short and long storing? So the long storing individuals were basically identified as staying sound in storage from harvest, which is September, October, until discard and cleaning, which was April, May. And so not as long as apples can be stored, but the short storing individuals all senesced within 3 months of harvest. And so there was a pretty clear separation between those two different types.
And what's the storage temperature?
It was kept at 4C, so pretty standard. Oh, that question was just what was the storage temperature? Ariel?
Just out of curiosity, this is really cool. Did you taste any of the apples?
Oh yes, I tasted almost all of them. Some of them are awful and hurt your mouth to consume, as Hungray can attest to. And some are actually quite pleasant with pretty good texture, sweetness, and acidity that, you know, is very similar to a commercial apple. But yeah, some are very high tannic and high acidity, and so they hurt. Any questions from Geneva?
Yes, lot of questions, just comments.
Okay.
Regarding the ML1, I think you can look at their genetic effect on their inter-allele interactions. For ML1, for both ML1 and ML3, they are dominant gene for high acid. So in your population, you have a homologous high acid for what? New York CO1?
Yep, for CO1. Yep.
Yeah. The other parent is you have a heterozygous, right?
Right. Yep. Heterozygote.
So that means your population would not surrogate for ML1 in terms of their acid levels that are controlled by ML1. So that's why you don't see it. You don't see the effect of ML1.
Okay. So, I mean, you're saying that because of the dominant acid allele that can come from either parents, that we would not see a difference in expression?
Right. Not expression, just their functionality things.
Okay. Yeah, we could potentially do an allele expression difference with those.
Yeah, for the MA3, that's the perfect because you have both of them, they are heterozygous. So you see them surrogate, then you see the effect.
Yep.
So that's explain your data very well. Good job.
Thank you.
Okay, we're about out of time. If you'd like to hang around, and I'm sure there's more things back there to eat. So thank you all very much.
This has been a production of Cornell University, on the web at cornell.edu.